Hello dear aspirants, welcome to GDC online classes and especially welcome you for my cognition sessions. Yes, my dear aspirants, we are discussing, I think you all of you know that we are discussing indole alkaloids as a part of alkaloidal alkaloids classes. So we have covered up to now, we have covered indole alkaloids. In that indole alkaloids, we have covered the ergot. Then we have seen that ergot life cycle, ergot the source and the chemical constants and the chemical dash for the ergot we have covered. The important parameters, important aspects along with some microscopy also we have covered. And now in this session we are going to cover the rest of the indole alkaloids. We all knew that. So the classification of these alkaloid will be based on the chemical structure. So based on the nucleus that they containing, because each nucleus, each structure will possessing a specific pharmacological action, a specific therapeutic action. So for that reason we are classifying them accordingly based on their structure, and based on the structure they are having some pharmacological action, and then based on the nucleus and based on the structure we are having some of the important drugs in the alkaloids, especially in indole alkaloids. Yes, we have covered that ergot, and now we are moving to that 𝙂𝙀𝙎𝙀 vomica. So now we can see right over here the 𝙂𝙀𝙎 vomica is a seed. So we all knew that. So the alkaloids, any of the secondary metabolites can be obtained from different parts of the body or different parts of the plant. So either maybe from root, stem, flower, seed, leaf, or rhizome, or maybe any of the seed or fruit. Yes, like how in this 𝙂𝙀𝙎 vomica, it is a seed. It is a seed, dried ripened seed, where we can get the strychnine, brucin. Such type of chemical constants are obtained from the 𝙂𝙀𝙎 vomica. Now we can see that synonyms: 𝘾𝙀𝙉 and dog poison. Yes, so you should definitely remember with respect to synonym. I said so many times. Meanwhile, I'm explaining each and every alkaloid. I'm only stressing mainly on the synonyms because most of the drugs they are asking about the synonyms. If there are any special names are there like 𝘾𝙀𝙉, we know the fig, right? Fig, that's a fruit. So like how it is in earlier stage, you can see right over here. There's a 𝘾𝙀𝙉 or 𝙂𝙀𝙎 vomica. So this is a plant, this is a tree, the 𝙂𝙀𝙎 vomica tree, and after the dried ripened seeds will be a compressed, a disc-shaped seeds are there. So these are the 𝙂𝙀𝙎 vomica seeds. So they are containing a lignified trichomes. Okay, so these are the unripened seeds. So from that fruits we can get these seeds. Okay, yes. Dog poison and 𝘾𝙀𝙉. Dog poison and 𝘾𝙀𝙉. Okay. Yes. And 𝙂𝙀𝙎 vomica, it is just coming to the source of this 𝙂𝙀𝙎 vomica. It is obtained from the dried ripened seed of Strychnus 𝙂𝙀𝙎 vomica. Strychnus 𝙂𝙀𝙎 vomica belong to the family 𝘿𝙄𝙎𝙎. Belong to the family 𝘿𝙄𝙎𝙎, my dear aspirants. Okay. So I said that so many times in this cognosy, it has a very important and very interesting fact that our drugs, the pharmacognosy, in that we are having several drugs. The drugs itself having the interlink between all the synonym, chemical constants, and the chemical rates and some other uses. Okay, so that's belong to some are maybe related with their family. Okay, so like how we're having several drugs like Vinka rosea is a source. Vinka is a common name, and Vinka rosea source. Okay, and whereas aposane is a family, and whereas 𝘂𝙀𝙎, 𝘂𝑏 are the chemical constants. So there is the resemblance. Meanwhile, they're pronouncing and their words. Okay, so like how that's what I'm trying to say is Strychnus 𝙂𝙀𝙎 vomica. So that strychnine will be obtained from the Strychnus 𝙂𝙀𝙎 vomica, that is from 𝙂𝙀𝙎 vomica seed. So belong to the family 𝘿𝙄𝙎𝙎. So most of the seeds, when we look at that microscopy, these seeds will be greenish grain color and disc shaped. These are in our earlier stages, these are completely ripened or dried ripened seeds. But when we look at the microscopy, the seed will be like in a greenish gray too. Like this is how disc shaped. So like how they are in circular, right? And bitter in taste, strychnine, brucin, they are bitter principles of the Strychnus 𝙂𝙀𝙎 vomica. And when you look at this image of the Strychnus 𝙂𝙀𝙎 vomica seed, it has a margin which is the outermost layer, and the centermost part is hilum will be there, which is a connecting part, and hairy surface which has the most lignified trichomes will be seen in this 𝙂𝙀𝙎 vomica. So these are the characteristic features. So this Strychnus 𝙂𝙀𝙎 vomica, or these lignified trichomes are the characteristic features of the 𝙂𝙀𝘵𝐾𝙀𝑒 seed, and they consisting of some of the ridges. You can see right over here. So this is consisting of the grayish matter. The grayish color will be because of the trichomes. Okay, so and it has some micropyle. It has some micropyle, my dear students. And now moving to that, when you look at the in detail microscopy of this 𝙂𝙀𝘵𝐾𝙀𝑒, you can see that 𝙂𝙀𝘵𝐾𝙀𝑒 transverse section of that seed, these are the lignified trichomes. That's what I said. So these are the characteristic features. If you want to detect the adulteration, if you want to confirm the identity of the 𝙂𝙀𝘵𝐾𝙀𝑒, even it is in a powder form. So when you need to confirm, when you need to detect, when you need to identify the particular 𝙂𝙀𝘵𝐾𝙀𝑒, when it is in a powder form, then immediately you should definitely go for the one of the characteristic feature, that is a lignified trichome. Okay, so in order to detect that 𝙂𝙀𝘵𝐾𝙀𝑒 when it is in a powder form, you need to check that when you place under the microscope, if you look at or if you detect any of the lignified trichomes, then you can confirm that is as a 𝙂𝙀𝘵𝐾𝙀𝑒. Even though, so we have an experiment in our pharmacognosy, one that is a length and width of 𝙂𝙀𝘵𝐾 trichomes, because even though so many plants are having trichomes, so many leaves are having trichomes, but what will be the differentiation between the what is the unique nature of these 𝙂𝙀𝘵𝐾 seeds and the other type of trichomes? They are having very larger, 12 micrometers length will be there, around 12 micrometers, and about 4 to 5 micrometers of the diameter will be there. So if you look at, in the powder, we are assuming, and we got a powder that is assumed as a 𝙂𝙀𝘵𝐾, but you need to confirm whether it is 𝙂𝙀𝘵𝐾 or not. So that if you place a particular powder under the microscope, and if you place it, and if you place some of the fluoroglycinol, and if you look at under the microscope, if you observe and if you measure the diameter with the help of the stage micrometer, okay, so that if the diameter and the length and the requirements and the units will be within the standard, so then will be confirmed. So this is about just for the quantitative microscopy, just to know about the practical knowledge. Okay, so these are the characteristic features of the 𝙂𝙀𝘵𝐾. So we can see that other microscopical characteristics are epidermis consisting of strongly thickened and pitted lignified trichomes. That's what I said, strongly lignified, thickened, pitted trichomes are there. So these are the characteristic features, and followed by collapsed cells. So these are the collapsed parenchyma, and this one is the epidermis, and thereby endosperm, and thereby somewhere in between these endosperm cells there are aleurone grains are there. So and in between that endosperm, the oil globules also there. Yes, that's what I said. So aleurone grains and oil globules are there. So this is about the microscopy, and you can see the chemical constants. That's what I said, strictness 𝙂𝙀𝘵𝐾. So that's the beauty of the cognosy, which means that the synonym and the species name itself having their extension, or is there having their chemical constants name, because we are having several or n number of drugs in the universe, in the entire planet. But how can you remember if you have a different names which are not at all matching with their genus species? So how can you remember? So for that reason, almost all chemical constants names are having a resemblance, or they are directly derived from the genus species name. So strictness 𝙂𝙀𝘵𝐾, strychnine. Okay, so coming to the having main two principles, that is strychnine, which is a more active and less bitter, more active form, therapeutically active, and whereas a brucin is a less active and more bitter form. Okay, so these are the main two key principles of this 𝙂𝙀𝘵𝐾. But apart from that, there are some other iron glands are there, aleurone grains are there. Okay, some other things, some other chemical constants there. But apart from that, it also consists of fluorogenic acid and glycoside, that is Loganaceae family. That's the reason loganin, that's the glycoside of this particular strychnine 𝙂𝙀𝘵𝐾. Sometimes you may get a question like, so the strictness 𝙂𝙀𝘵𝐾 consisting of which of the alkaloid, which of the glycoside? Because we are discussing this strictness 𝙂𝙀𝘵𝐾 or 𝙂𝙀𝘵𝐾 seed in the part of alkaloid, especially in the indole alkaloid. But even though some alkaloids, we can't consume, we can't confirm that only one plant is having only one type of secondary metabolite. Even as like our body organs, we are having heart, we are having liver, we are having digestive system, we are having skeleton, all organs having their special function. Okay, like how even though the plants are having almost all 80% of alkaloids, 80% of all secondary metals, the plant may consisting of alkaloids, may consist of glycosides, resins, volatiloids. But how can we classify a drug into a specific section of secondary metabolite? Like how can you confirm a drug into alkaloidal drug? How can you confirm a drug into glycosidal drug? Because of that, the majority or amount, if it is if the drug consisting of higher amounts of alkaloids, it can be considered as an alkaloid drug. It is consisting of glycosides, glycosidal drug. So like how we classify the drugs, okay, but I not confirm that the particular drug is having only one type of secondary metabolite. They may also contain other than the alkaloids, other than the glycosides. Yeah, that's my point. So that what I mean to say is, apart from that, strychnine, brucin, there is also another chemical compound that is called loganin, which is a glycoside, like, okay, and chlorogenic acid is also there. Is that clear, my dear aspirants? That what should you know? And now moving to that, uses of this particular strychnine 𝙂𝙀𝙀 or 𝙂𝙀𝙀 acid. So due to its bitter, this is used as a bitter and stomachache and tonic. But almost all alkaloids, 80% or 60 to 80%, we can consider them as a CNS stimulants. Okay, because they are alkaloids, they are most of the alkaloids having CNS. They are acting on the entire CNS. Either maybe some of they producing the sedative effect, some of CNS depressants, or some are CNS stimulants. Like if you take an example of any other alkaloid with respect to CNS depressant property, sedation property, opium is the best example for the alkaloid which causes sedation, which causes CNS depressant. But whereas caffeine and like how this 𝙂𝙀𝙀, they are CNS stimulants. Okay, and it stimulates the respiratory system and cardiovascular system because it is CNS stimulant. If you alter the function of the CNS function, thereby ultimately if you activate your brain, then brain activates all other channels which are interconnected inside our body. So if the brain function is very high, you can got adrenal rush, thereby your heart rate is increased. If your heart is get increased, okay, thereby the the entire process, the respiration, all these are get enhanced. So like how obviously the stimulates the respiratory system and cardiovascular system. So it has a functions on because it also having one of the glycoside, that is loganin. So that reason it is some cardiovascular system. So coming to that, what are the adulterants or substituents? Strychnine potatorum, that is India also known as clearing nuts. So which are very resemble, just similar to that of the 𝙂𝙀𝙀. So that in India, so strychnine potatorum is a drug where the people used to substitute the strychnine 𝙂𝙀𝙀. And strychnine 𝘋𝑗𝑠 in Burma it is available. So it contains no strychnine, but it has a it doesn't consist of any strychnine or brucin. Okay, so sometimes they ask about the adulterants or substituents. You definitely remember that strychnine potatorum and strychnine 𝘋𝑗𝑠. And whereas the allied drugs are like strychnine 𝘂𝙂𝙎𝑎𝑛𝑎, strychnine 𝘂𝙂𝙎𝑎𝑛𝑎 and strychnine 𝘂𝙂𝙎𝑎𝑛𝑎. So these are about the allied drugs, adulterants, and uses on some of the important factors or aspects of strychnine 𝙂𝙀𝙀. Okay, so this is about the 𝙂𝙀𝙀 acid. And next we will see the other drug that is called Rauwolfia. Can anybody guess, my dear students, Rauwolfia? So we all knew that the Rauwolfia is consisting of one of the important alkaloid called riserpine. Rauwolfia serpentine. It is known as sarpagandha. I said you so many times, if the particular drug is having a special synonym, sometimes they denote their pharmacological action. Like how in the deadly nightshade leaf of the Atropa belladonna, and like how sarpagandha, which means sarpagandha or in a snake root. The name is saying that sarpa refers to snake, gandha means medicine. Snake, it's an antidote for the snake poison. So because of that particular pharmacological action, it got that name of a synonym, that is sarpagandha or snake root or chota chand and devil pepper. So this is a Rauwolfia. So we can see the leaf, you can see the image of this particular Rauwolfia. So it is a very familiar, it can be seen, abundantly seen in our localities itself. Okay, so it can be grown in almost all type of conditions. Okay, so this will be very familiar plant for all of us, but we never knew that is a Rauwolfia. But we have seen that flowers and we have seen that leaves. but we never knew the importance of this particular plant. Okay, so that coming to that, the source of this particular Rauwolfia, it is obtained from rhizomes and roots, so especially from the subterranean organs. So I'll ask you one thing. So why so what it means? So some of the plants are having the alkaloids in the leaves, some of the alkaloids may having the alkaloids in seeds, so some of the alkaloids having synthesized in roots. Why? Because their site of synthesis, and the elements and the other micro and macro nutrients required for the metabolism and synthesis of secondary metabolites or specific secondary metabolite will be favorable at that specific location, specific part. So for that reason, they will be seen in a specific parts like roots. If they are synthesized in roots, so that favorable condition for the synthesis and metabolism will be in roots. If they are metabolized, if they are synthesized in the leaves, so that will be then that favorable condition. That's what it meant. Okay, a simple IQ. So it will be obtained from Rauwolfia serpentina. I said that there is a interesting fact that there's the chemical constant is the main chief constant here is reserpine. Okay, serpentine. Okay, serpine, and from this R, reserpine, and other drugs like serpentine is also there. Reserpine. Okay, we'll see the rest of the things. So coming to the family, it is an Apocynaceae. The family Apocynaceae. And coming to the microscopy. So if you look at this, because it is when we talk about the microscopy, you should definitely think of the part which was mentioned in the biological source. So here, what is the part? Root or rhizome. Root or rhizome will be the we are going to see that because at that part only, at that particular site only, we got that reserpine. So that we should think of, we should definitely look at into that microscopy of the root in rhizome. So if you look at the TS section of root, shows externally striated cork. So this is a cork. My dear aspirants, I'll tell you that as a cognosy professor, I'll tell you that. So if you never remember that a specific parts of the particular plant or particular part, okay, the microscopical section or microscopical parts of the particular thing, you should definitely think some common points or some common parts will be there. Like if you check about the leaf, so epidermis, okay, margin, leaf, base, apex, vein, okay, parenchyma cells, stomata. So like how they are common. So like how in almost all barks or roots and rhizomes, bark, cork, combium, phylloderm. These will be the common factors, common parts of these things. Okay, so that you should definitely think of that. Okay, so whenever you thought of that bark or root or rhizome, so your mind should definitely remember that it is consisting of our outermost layer, that is a cork, and followed by phylloderm, and followed by phloem. And if it is a bark, and there will be combium. Okay, combium will be there. So like how you need to remember, that's a trick. That's a very basic parameter you should remember. So it consisting of striated cork with 2 to 7 layers. It is, the thick one, look here. So it is consisting of 2 to 7 layers, even though it is a very small diagrammatically here we represent it very small. So but it is 2 to 7 layers of small cells followed by phylloderm, and there will be a medullary race also there, phloem fibers. Okay, and xylem is meant for the water supply and phloem is meant for the nutrients and other supply. Because it's a root and rhizome, from that root and rhizome the absorbed in the nutrients and all will be supplied to the stem, thereby transferred to the all of the rest of the parts. So phloem will be there definitely, the medullary race, vessels, growth rings. So by that growth rings we can assess, we can expect, we can calculate the age of the particular plant, and fruit of xylem. Okay, and both bark and wood contains abundant starch, because you should remember that if any part which dies down towards the earth, maybe any of the subterranean part definitely consisting of starch. You should definitely remember. I'll tell you best example. Does I think potato consisting of starch, right? And any of the sweet potato, colocasia. So subterranean organs, all maybe root, rhizome, or any other subterranean organ will be consisting of starch as an additional component. Okay, and xylem is entirely lignified. The xylem, protoxylem, sclerenchyma is absent, and tetraschious arrangement will be present. Okay, so this will be the microscopy of Rauwolfia. and we'll see the chemical constants of this Rauwolfia. I said that from the reserpine, from the Rauwolfia serpentine, we got the name itself denoting that reserpine. This is chief constant. Resinamine, 𝙂𝙂𝙎𝙬, 𝙂𝙂𝙎𝙬, serpentine, resinamine, serpentinine, and 𝘂𝙂𝙎𝙬. So these will be the chemical constants, but among all, reserpine will be the active one, which is having a more active value, more therapeutical value, and rest of the things will be synthesized, and further they are having for other pharmacological actions. We will see. Rauwolfia, especially this Rauwolfia, is an anti-hypertensive drug, anti-hypertensive in nature. So it is used to treat the BP, blood pressure, and used in a neuropsychiatric disorder. So I said that every alkaloid, either maybe only or partially, maybe half of the that maybe consisting of partially they are acting on CNS, or completely they are having a CNS action. So like a neuropsychotic disorder. So it is true neuropsychiatric disorder. And 𝙂𝙂𝙎𝙬 used in treatment of arrhythmia. So maybe it's consisting of some of the cardiac properties. And 𝙂𝙂𝙎𝙬 used in circulatory disease and relief of obstruction to the normal cerebral blood flow. When it any other obstruction leads to the cerebral blood flow, such conditions also we can use the 𝙂𝙂𝙎𝙬. 𝙂𝙂𝙎𝙬 is for arrhythmia, arrhythmia, 𝙂𝙂𝙎𝙬, and 𝙂𝙂𝙎𝙬, cerebral blood flow. 𝙂𝙂𝙎𝙬, C, cerebral blood flow. 𝙂𝙂𝙎𝙬, arrhythmic, A, because it is having other extension like C, cerebral blood flow, like how you need to remember. Okay, yes. So these are the uses of Rauwolfia, and so that's about Rauwolfia. Next, we'll see the one of the other important alkaloid, which is very familiar for their anti-cancer property. We well known that whenever I thought, maybe you never thought about, whenever you heard about the Indole alkaloid, maybe your mind will remember some other drugs. But according to my knowledge, whenever I heard about the Indole alkaloid, definitely my mind will goes to the Vinka, because the Vinka is having about 120, about 100 plus types of Indole alkaloids will be there. But among them, only 2 are very important. Only 5 are important. That is 𝘂𝙂𝙎𝙬𝙬, 𝘂𝙂𝙬𝙬, 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 okay, 𝘂𝙂𝙬𝙬. So these are the main 5 active constants or 5 active alkaloids, Indole alkaloids, which are therapeutical active and shows a prominent value in medicinal field. So we'll see. It is the synonym, we know that. So it will be available in so many types of species. The Vinka will be having a different color, different species will be there. But only the anti-cancer properties, especially the 𝘂𝙂𝙎𝙬 and 𝘂𝙂𝙬𝙬 will be obtained from this species called 𝘾𝙀𝙀𝙀 or Vinka-rosea. 𝘾𝙀𝙀𝙀 or Vinka-rosea. So these are purple color. Purple colors will be there, flowers will be there, red color will be there, white will be there, blue will be there. So there are different species. But so it is known as the synonym. You should definitely remember that. Okay, I'll tell you a hint that what is the color of this plant? Flowers, purple. Purple periwinkle. Purple periwinkle. Okay, so the synonym of the plant is. So whenever you heard of the Vinka, you should definitely remember this image. So in that image, the flowers are in purple color. So purple periwinkle. So that periwinkle, purple color. And Sadabar puli, Madagascar periwinkle. So these are the synonyms, because sometimes we may include that there's geographical source, because a Madagascar region will be more favorable for their growth. So Madagascar periwinkle, Sadapuli. Okay, so like how. And source, it's obtained from the dried whole plant. So up to now we have seen only a specific part will be consisting of the chemical content, but when coming to this Vinka, it is consisting of entire whole plant is having the alkaloids. We can take whatever the part of the plant. You can take the root or stem or any part of flower, maybe anything. Okay. And coming to the family Apocynaceae. Yes, coming to that. So for Vinka, the TS of the Vinka will be an important thing. Sometimes, because some plants have some of important plants like Datura, Vinka, Nux vomica. So simple. what are the microscopy we have included in these sessions, in these classes. They are very important because from those microscopies or TS, so many questions are given in our previous GPAT examination. So that whatever we included here, please go through it, do remember, do highlight the parts, so that it will be very helpful for you, because there is a chance of getting questions from the TS or microscopy also. Okay, we'll see the microscopy. So when you look at the leaf TS, it is a dorsal part, which is upper part, and ventral part, this is lower part. Okay, so we can see the 𝘂𝙬𝙬𝙬 and the lower epidermis. Okay, and palisade parenchyma will be there, upper epidermis will be there, and consisting of covering trichomes. Most this is consisting of covering trichomes. Sponge parenchyma will be there, and xylem and phloem will be there. Xylem and phloem will be here. Okay, so these are the common or main important parts. It's consisting of all the parts, but what special feature is consisting of 𝘂𝙬𝙬𝙬 and the covering trichome. CC, Vinka. Okay, CC. We'll see. Upper epidermis shows the presence of single layer of rectangular celled epidermis. So there is epidermis, rectangular cells. You can see the rectangular cell epidermis here. Okay, and with the unicellular covering trichome. So unicellular covering trichomes, you can see the covering trichome here, you can covering trichome here. These are unicellular, only single cell will be grown as a covering trichome, and consisting of sponge parenchyma. So will be like this, like how like this. There will be sponge parenchyma will be there, and cruciferous stomata or anisocytic stomata will be there. You should definitely remember that what type of stomata will be there. Okay, and what type of trichome will be there. So stomata, Vinka is ends with A, anisocytic. Vinca rose S. Okay, anisocytic, because I'll tell you a hint that Vinka ends with, spelling ends with A. So it consists of anisocytic stomata. But how can you remember that? Because anamocytic will be there, anisocytic will be there, actinocytic also there. But when you look at that hint, another hint in this, S, Vinca rosea consists of S. So anisocytic Vinca rosea. Okay, I think you understood my trick. Even if you're not able to understand that, leave it aside. Just do remember that anisocytic stomata will be there. And coming to the main important thing is chemical constants. In this chemical constant, there are about, I said that 100 plus, more than 100 type of alkaloids, indole alkaloids are there, but where the very important or chief chemical constants are vincristine and vinblastin, which are anti-neoplastic agents or anti-cancer agents. So they had a special place in the cancer treatment. Especially they had a very immense role in the treating the cancer, almost all type of cancers. Okay, so vincristine and vinblastin. And whereas, and it also consisting of a part called vinblastin contain indole part called catharanthus roseus, and dihydroindole alkaloid. So apart from that, it is having vincristine, vinblastin, vindolin, vinclozidine, vinlurocin. So these are things, and apart from that, it is also consisting 𝘎𝑒𝑗𝑒𝑚𝑎LIN. Okay, so they're all built there. And coming to that, vinblastin. So when you extract the Vinca alkaloids, so higher amounts will be the vinblastin. You can get the higher amounts of vinblastin, but in order to get vincristine, you need to do the demethylation with the help of streptomyces 𝘎𝑒𝑗𝑒𝑚𝑎LIN. So then you'll get vincristine, which is superior activity. So initially only vinblastin will be there, as like the hyoscine in the atropa. So atropine is not directly available, but when the racemization takes place, atropine will be produced. So like from vinblastin, due to N-methylation, we got vincristine here. We know that anti-neoplastic agent, hot skin disease, and acute lymphocytic leukemia in child, and also it has a hypotensive and anti-diabetic action. So it has a very immense role. Because of this properties, a Vinca will be one of the it becomes one of the essential medicines list. It's added in the WHO essential medicine list because of this n number of health properties: hot skin disease, hot skin lymphoma, and acute lymphocytic leukemia, and such type of cancers can be treated by the Vinca. So this is about Vinca. And last in this indole alkaloid is physostigma. So it is somehow looks like a bean plant. You can look over here. So this is seed of the phytostigma. So these are the plant, entire plant, herb and entire herb. Okay, so this will be that seed of bean. These are the tendrils, and these are the other parts of the part of the phytostigma. So it is known as calabar beans because it is beans, calabar beans. So that the name itself saying that what will be the chemical quantities phytostigma, phytostigmin. But coming to the source of this biological source of the phytostigma will be obtained from the ripened seeds. These are the seeds of Phytostigma venenosum. Phytostigma venenosum. Physostigma venenosum, not Phytostigma. Physostigma venenosum. Belong to family Leguminaceae. Leguminaceae. Okay. And the chemical constants. We have included only the important things here. Apart from that, they are not that much important. Apart from these things. So chemical constants are phytostigmin, that is isorin, isorylin, isoramine. Okay, isorin, the phytostigmin known as isorin, isorylin, and isoramine. But from this phytostigmin, we can get two other products, that is from the phytostigmin, that is isorin. By the hydrolysis, we can get isorylin, and by up to and because of a light exposure, we can get rubeserin. Okay, rubeserin. So these are the main active constituents of the physostigmin, and whereas it is an anticholinergic agent, so it means they are acting on seizures and also some of the muscle contractions, spasms, like how in all these things on all type of diseases we can use this phytostigmin. Okay, and used in ophthalmology for contracting a pupil of eye to counteract the poison caused by the anticholinergic agent. So in the ophthalmology, especially to treat the construction of pupil of eye because of some of the contract caused by the anticholinergic agent. Yes, my dear aspirants, this is about the indole alkaloids. We'll see in the next upcoming session with a new another type of classification, next alkaloids. Is that clear, my dear students? Yes, we'll see in the next class with a new another alkaloid. Thank you. Have a great day.